Recording in progress.
Good evening. It's my immense pleasure to welcome you to this evening's webinar. I'm Sabina Dizdarevic, your current BNMS president, consultant in nuclear medicine, professor of nuclear medicine, University Hospitals, Sussex, NHS Foundation Trust, and Brighton and Sussex Medical School. Very warm welcome to all of you, and thank you for being here. I would like to take this opportunity to thank to our sponsors, Siemens Healthineers and Novartis, for their support and collaboration. Tonight, we are honored to be joined by three esteemed past presidents of BNMS who will share their experiences during their presidencies, challenges they faced, the inspirations that shaped their leadership, and the achievements that marked their terms. They will also reflect on the future of our specialty and how, together with our members and in collaboration with national and international colleagues across societies and colleges, we can continue to shape the direction of nuclear medicine. After the interview with past presidents, you will all have an opportunity to ask questions. This is the first in a series of webinars bringing past presidents into conversation with the current president and new members of BNMS and other delegates, exploring the past, present, and future of our field. This year, we are celebrating the 60th anniversary of BNMS with a series of events planned in the lead-up to our annual meeting and continuing afterwards, culminating on the 19th of July, the date on which the society was founded in 1966. Development of radionuclide techniques for diagnosis and therapy has a long and distinguished history in the UK. Our society was founded in 1966 in a friendly, vibrant atmosphere in Prince Alfred Pub in London. Among those present were Dr. David Keeling, who is with us this evening, along with Professor Ralph McCready, Steve Garnett, and late Edward Williams, the founding members. The society was created to provide a platform for regular discussion of emerging techniques, instrumentation, staffing issues, and to promote nuclear medicine. The first meeting took place at Middlesex Hospital in London, followed by a buffet supper. At that time, it was agreed that 4 meetings would be held each year. Today, we continue that tradition through 2 national meetings and multiple face-to-face regional and local events, and online webinars and seminars across the UK. It's now my pleasure to introduce our speakers. This evening, we are joined by Dr. David Keeling, Professor Sobhan Vinjamuri, and Professor John Buscombe. Let me begin by introducing Dr. David Keeling, one of the pioneers of our specialty and the founding member of what became the British Nuclear Medicine Society. David was president of BNMS from 1994 to 1996. David qualified at Cambridge and Middlesex Hospital, and after his early general medical training, he sought a career in what he described as scientific medicine. Interestingly, he initially trained in pathology, but soon realized he wanted more direct patient contact. In 1962, the Middlesex Hospital needed hematologically trained support for the emerging field of nuclear medicine, and this became David's entry into the specialty. It was an extraordinary period of innovation. We never stop innovating. Even then, David was involved in developing early to-do-it-yourself radiopharmaceuticals and of in-house instrumentation, including work on the first tomographic scanner in Europe. Small group of 4 clinicians working in this exciting environment went on to form the society that would become the British Nuclear Medicine Society. At that time, long before ultrasound, CT or MRI, nuclear medicine already offered a remarkable broad range of clinical applications and operated with far fewer regulatory constraints than today. David also established the UK Adverse Reaction Reporting Scheme for radiopharmaceuticals, and he will tell us more about that. In 1973, he established full nuclear medicine service for Plymouth and Cornwall, a role he found deeply rewarding, combining scientific innovation with meaningful patient care. Although he retired from NHS in 1995, he continued to contribute to the field by chairing very active independent Phase 1 ethics committee until 2009, as he puts it, finally becoming a poacher turned gamekeeper. David, can you please then join us for this evening, and very warm welcome to you. Hello, David.
Good evening. Thank you.
So I just wanted to ask you, looking back, what were the key challenges in nuclear medicine during your time in NHS and during your presidency, and your reflection on that time in 1996 and onwards?
Well, there's a big gap between starting the society 60 years ago and when I came president, which is only 30 odd years ago. In fact, I retired from the NHS during my presidency, but I had started in what has become nuclear medicine when I was a pathologist. I was a hematologist, and by some extraordinary coincidence, this bottle of iron 55 ferrosuccinate, iron 50, was my first experience. As a chemical pathologist, we had to sort out, we were looking at iron absorption studies, 55 and 59. But the 55 is a difficult one to measure. It's liquid scintillation counting of a small energy beta. And so it was I, as a chemist, that started doing that and moved over to nuclear medicine to do it. And it was good fun. And technetium hadn't arrived, but we were using a small range of odd isotopes, arsenic, mercury, you name it. Iodine, of course, was the main clinical one. But it brought me back in contact with patients, which is something I had realized that if I went into pure pathology, in those days, pathologists had very little clinical contact. And when, after a year, the recall was sounded by the pathology department, I said goodbye to them and stayed with Edward Williams and ran the clinical branch of nuclear medicine at the Middlesex.
It's amazing, really. I think that's what really brought us, many of us, to nuclear medicine, that contact with the patients, because we are imagers, but we do like to see our patients and treat patients, and all this amazing field of development of diagnostics that we will be talking later on, both John and Sobhan, is something which is still emerging and really putting patients in the heart of everything what we do. So during your presidency, because you said it was a long time, is it since you started and then you became president, although you were founding member, but it took you a long time to become president, which I think is great with all your experience that you brought. Did you have anything particular in that decade, like during your presidency, that you could reflect, and any challenge that you can tell us about?
The main thing that happened when I was president, my first year we were meeting at Imperial College, as we had been meeting for some years. But the expense was proving a bit much for our treasurer, and it was undermining our financial stability. And we decided to move out of London. And I, with the help of colleagues, we picked Brighton as a place that was offering us a good conference venue at a price we thought we could afford. So we moved out, and I think I only upset a few of my London-based colleagues who weren't on their doorstep for the meeting. But everyone else seemed to enjoy it, and Brighton looked after us well. And as you see, the rest is history. We've moved around ever since.
Yeah, all roads are leading to Brighton, including mine as well. So mine was the first conference in Brighton, and clearly I've been consultant in Brighton now for more than 20 years. And I'm sorry that meeting is no longer in Brighton, but I hope we will have at least autumn meeting soon, some point in Brighton. Yeah. I think the main thing about Brighton was that industry wasn't very happy, because after many years of establishment, the conference in Brighton, people got used to Brighton so well that they were going out to restaurants rather to the conference center. So we started rotating outside of the Brighton, and as you all know, our next conference is in Manchester. And I think it's working very well, taking a meeting outside of London, not being only London-centric, worked very well. And thank you for that, David. It was something which was a major step. And obviously, you encountered a lot of problems with that, but you've done extremely well. So with regards to your role of president, what actually inspired you to take on that role?
I think I got pushed from behind. I'd come down to Plymouth and looking after Plymouth and Cornwall back in 1973. And I was single-handed with no junior staff. So I was racing to Truro twice a week, which kept me quite busy. But it also meant that we had left a small semi-detached house in London at 1973 prices. And with not much added, I bought a 6-bedroom house where I'm today.
Wow.
So, with 4 children, 4 young children, it was a good family move.
It was an excellent move, obviously. Oh, well done, David. It's just amazing. So, and having 4 kids, I think it's major achievement as well. Probably something that you are very proud of, the top of your medical career.
They're all at peace with each other. In fact, we all had Christmas together, so the family has done well.
That's really amazing, really amazing. One of your achievements were to open a registry for like adverse events. Can you tell us a little bit about that?
Well, we started finding, you remember in the early days, you did not buy all the radiopharmaceuticals. In fact, there was a limited selection and quite a lot of it was do it yourself. And if something went wrong, these were usually minor things, but people were not necessarily too keen on telling the world at large that they'd dropped a brick. And so we started an informal setup because we all knew each other. That was the real thing. The numbers were small and we all knew each other. And people were prepared to send details to me on the understanding that when it went further, it would have been anonymized. And it worked. People were interested in doing it and learning from it. And subsequently, when Brussels started wanting to bring in legal things about radiopharmaceuticals, I was invited to Brussels to make some suggestions. I don't know that they liked them, being told that the doctors were better looking after it themselves and letting the bureaucrats interfere. But the only people reporting adverse reactions were the British and the East German. The East German had been persuaded one way or another to line up and do their jobs and report everything. And we were the only 2 countries who appeared to have any adverse reactions. Clearly, the others just ignored them. But it's worked. We made a lot of discoveries particularly with the semi-homemade preparations. I do remember one of my physics colleagues had got the gelatin for a radiopharmaceutical from a shop.
Oh, couldn't happen nowadays, definitely.
And so yes, we had difficulties, particularly for the do-it-yourselfers.
Yeah, and like Lily, but you know the registry is working very strong nowadays, and Maggie Cooper, she's like on the top of leading in that area. So I think it's really important to have structured system like that, is it? Sometimes we might think we went too far in a way, with a lot of regulations, as you mentioned as well, describing your CV. That time when you were working, there were far fewer regulatory constraints than today. So is there anything like more that you can tell us about that?
Well, when gallium started getting into the news, it was being produced at Hammersmith, the cyclotron unit, and they were not prepared to share it with anyone else. But one of my technicians went to work for Philips in Petton, and he rang me back and said, "Did I want some gallium?" I said, "You know jolly well I do." Um, and my first delivery, we had taken some rats with a sarcoma, in fact, and injected them, and it shone out like a lighthouse. The next week, it went into me, and my wife was taking blood during the night to give us the blood clearance and the excretion data. And the next week it went into a patient, having explained to him that I'd tried it last week and it had done me no harm.
Yeah.
And an unknown primary in his esophagus shone out like a beam. And being an assistant editor of the British Journal of Cancer, it was in print before Hammersmith knew what was happening.
Wow. I mean, these are the stories that currently, we can't comprehend, but without bravery of many people, probably we wouldn't be here nowadays. Although it's much more structured and we have a much more opportunities for research, but sometimes we do have difficulties to accessing trials or developing trials. We do know we do need to have regulations, as you rightly said. It's important, and you established one of the systems for monitoring side effects. But sometimes we know that, particularly in the UK, a lot of trials are delayed nowadays because of enormous amount of bureaucracy that we are doing. And we're trying to talk to MHRA about that and to try to streamline. Clearly, I think these stories I heard before from some other people, like who were like of injecting themselves and trying on themselves. I think nowadays, any of us would probably try to do it. But to be honest, when I was doing my PhD, but although it was ethically approved, I also did my genotyping myself for saliva expression of the PGP, ABC proteins, but fully ethically approved and approved for the normal volunteers as well.
When we first had technetium, we had been jumping from microcurie doses to millicurie doses almost overnight. And I do remember our sister, the nursing sister in charge of our nursing staff, becoming exceedingly worried about the radiation dose they were going to get. And we did a lot of experiments on ourselves with doses to look at the physiology of it and the thyroid uptake and so forth, and then also to measure the dosimetry around the body and where to stand and where not to stand next to a patient full of millicuries of technetium, even though the doses in those days, I mean, the generators were only sort of 10 millicurie dose generators, not like the ones we have today. But these things, the obvious thing was to practice on yourself.
Yeah, definitely. So, David, what's about your most memorable moment of, your time with BNMS? Is there anything that you are particularly proud of? Clearly, you already mentioned so many things, and I don't know whether you can think about anything else, but...
I think the main thing was making peace with the physicists.
Oh, that's really good one.
In the early days, a lot of physicists had been playing amateur doctors. And I mean,
Okay.
Edward Williams had been the physicist at the Middlesex doing the iodine studies and had been told by Professor Windayer, who was in charge of virtually everything, that if he wanted to stay like that, he needed a medical degree. And so he went through medicine at the Middlesex and married one of the nurses, who turned out to be junior to the nurse I'd married. But that was the way of finding whether you needed a medical degree or a physics degree or a bit of both. And some of the medical physics-led departments were putting up the shutters and trying to keep us out. And the early days of the BNMS, you needed a medical degree to join it.
Yeah, that's what I read, that it was a primary medical society. And then clearly now with the all developments,
Yep.
we become
The
multi-professional
only
society. Thanks to you as well, is it partly in a way because you made a peace with the physicist, and we clearly have all our clinical scientists and technologists, radiographers, nurses now as members in the
when I
society.
started, there was only one radiopharmacist in the country. That was Mr. Little in Liverpool. Whenever the pharmacy department of Middlesex heard what we were doing, they didn't want to know anything about it at all. They thought it was the work of the devil, I think.
Wow. So, but clearly, I mean, now still struggling with workforce, but when you think what we had 60 years ago, it is a big change, and we did really develop dramatically, is it?
Yes.
And
Oh, yes. Yes.
yeah,
Yes.
so I think
Common sense has prevailed.
definitely. So I know we could talk to you all night, and I think we will also have a lot of questions from other presidents tonight to you directly at the end of their talks. And we'll move out now to Professor Sobhan Vinjamuri, who has different challenges during his presidency. As any president, he had opportunities to achieve, but also to find a way to overcome challenges. Professor Sobhan Vinjamuri is a lead consultant in nuclear medicine at Royal Liverpool University Hospital, role held since 1998. He has been honorary clinical professor at University of Liverpool since 2014. He has held numerous national and international leadership roles, chair of the Joint Specialty Committee on Nuclear Medicine at the Royal College of Physicians from April 2020. He is also currently member of the E&M Ethics Committee and serves as the editor of Nuclear Medicine Communication, position he has held since 2009. Professor Vinjamuri has extensive experience in governance and ethics, including membership of the Administration of Radioactive Substances Advisory Committee and chairman of the GM South Research Ethics Committee. So he will also talk tonight, and he will bring us a talk towards higher specificity, which brings together cutting-edge hybrid imaging, advanced diagnostics, and theranostics technologies alongside his personal journey through these developments. Hello, Sobhan. Welcome to the President webinar, and thank you for sharing your experience. So, as I said, each president had a different challenges, opportunities. What were your challenges during your presidency?
Well, everything pales into consideration when you got Brexit as your main challenge. So nothing else matters. So I had Brexit, and we had people all over the world, especially even in the UK Parliament, Royal Colleges, specialist societies worried about Brexit and the impact of what that would do for importation of radiopharmaceuticals, export of radiopharmaceuticals. So I think it was a really crazy time, if I can use that very nice technical term. But we needed to be cool and calm because we were secretly hoping as BNMS Council that industry would help, because it's in their commercial interest to make sure that things would work out. So, and hopefully things are a lot better now. It's not ideal where we are right now with our access to radiopharmaceuticals, but it is a lot better than when we were dealing with it actively in 2016.
Oh, what a challenge, honestly. And then just to follow on a COVID. So, yes. So basically, but you were still you still wanted to take on the role. And although maybe when you were taking that role, you didn't know that Brexit would happen, although it was emerging. So what inspired you to take on the role of the president?
So one of my basic philosophies is you have to be the change you want to see. You cannot, comment, criticize from outside and say, well, oh, this is always like this, isn't it? And so I have always believed that if something can be done by you, you should do it. It's it's easy to comment from outside. So the challenge I thought was, well, okay, if we can influence from inside rather than standing outside BNMS Council and BNMS and saying, yeah, they always do that, don't they? And so on. So I just thought, okay, this is something that I could actually see if I work from within and see if I can contribute.
Excellent. So, and is there any particular initiative of legacy that you are most proud of?
Well, at that time when I was there, we had this concept of the BNMS President's Medal, which was the only way that we could recognize big contributors to nuclear medicine. But when I was president and I was considering people, there were 4 or 5 people at that time. So I said to our council members at that time, why don't we recognize a few more people? So we instituted the BNMS Roll of Honor during my presidency, whereby we recognize more than one person, because as you know, nuclear medicine is such a multi-professional group and so many people contributing at various levels. If you're going to pick up just one person, it might be good for that person, but you're not being fair to everybody else. So I strongly believe that is one of the easy solutions that I could contribute to by recognizing more than one contributor to nuclear medicine on an annual basis. And I think it is still running very well, if I'm not mistaken. Is that right, Sabina?
Yeah, definitely running very well. And thank you for introducing additional roles of honor or additional awards, because it's really important for the members to be recognized, is it? And sometimes some people just inadvertently get missed and overlooked, particularly you think, oh, people, they had to have it, and then you just can't believe they didn't. And so that was great, Sobhan. And what was the most important development during your presidency then?
Well, I mean, I think from a memorable point, building up on what you have said before about missing people out, I feel it almost brings tears to my eyes, because I remember that was looking around and people might remember Muriel Buxton Thomas. So we issued the President's Medal, the letter to her while she was alive, but she had actually passed away by the time we actually awarded her physically. So her daughter came on her behalf and received it. And that was a very emotional moment for me to say, okay, and the daughter actually told us that, she actually knew that she got the BNMS President's Medal before she died. So that was actually a very moving and a memorable moment for me personally, because I felt that, like you said, some people do get missed out over time, and I felt that we need to recognize people in a big way. So that is one of my most memorable moments as BNMS president. And one more issue which I've used, legacy, if you can use that term, is I've introduced a new category of article on the nuclear medicine communications called BNMS communication. Because we are closely allied with the BNMS, why not have a special communication for BNMS where it's already peer reviewed? You don't need to spend extra time peer reviewing it, peer reviewing articles. If BNMS wants to rapidly communicate with its readership, to nuclear medicine communication readership, then that's another category that I've introduced. So I'm hoping that will work out well.
Thank you, Sobhan. It was really, really great work. And, for us who knew and who were friends with Muriel, that was something special. Thanks for that. And clearly communication as well. It's an important channel for BNMS to communicate with the members and with the world. So can you just share your talk with us, please? Because I think then you can talk through your personal journey and developments anyway, so that rather than through discussion with me.
Can you see my slides?
Yes, yes. Just on the slide mode, yeah.
Yeah, can you see that now?
Yes,
Okay.
very well.
Thank you. So I'm sorry if I'm looking a bit cockeyed at the moment because my camera is there and I'm looking there. So don't look at my eyes, just listen to my voice. So as you've said, I'm going to share my personal journey, but you also wanted a CME angle to it. So I've got a bit of science in it, a bit of education in it for the youngsters especially. So no surprises there, born, trained up to diploma in radiation medicine in India. But how did I come to the UK? You'll be surprised. At that time, we did not have emails. We did not have communication. We did not have LinkedIn. We did not have anything. So I physically wrote to 10 authors of the textbook of nuclear medicine at the time that was available in India, Britton and Macy. And guess what? One person responds. So it was Andrew Hilson who responded to my email, to my airmail, and said, "Come over. We'll look after you." So I came off to the UK. I was expecting to spend time with Andrew Hilson, but I think he was full or something. So I ended up with Keith Britton, and so that's my journey, beginning with Keith Britton in St. Bartholomew's Hospital, and then I became a trainee in the system. And one of the things which was crucial, which was actually asked as a quiz in the hospital where I worked in, it said, "Who's the youngest consultant there?" And they said, "Oh, at that time it was me." So a small claim to fame in my hospital. Real challenge is when you come from a different culture and you say, well, your professor at that time gives you an article to work on, and you do not know the meaning of it. You do not know what you're working on. And I had not realized that it was such a pivotal paper that he had given to me to work on my master's paper. So can you believe it? It's this paper on infecton imaging, which was actually published in the Lancet. So my first ever publication was in the Lancet, and thanks to Keith Britton, who gave this project to me as an MSc project. He just said, "Yeah, go and do some work on this, do some analysis, and you can get your name on the paper." And I said, wow. And so this is how sometimes you have to be in the right place at the right time, as they say. And the real interesting thing is, whenever I visited other places and students doing projects on imaging agents, infection imaging agents, when they turn out and say that this is this guy who actually published in the Lancet, because anybody who's doing any research work on infection imaging agents, this is one of the papers that they have to quote in their work. So it was quite interesting to see. I've seen students all over from India, from Turkey, from China contact me and say, can you share some insight? So my first paper was in the Lancet. And just to show an image from that paper where you're looking at a superiority to white cell scans. So white cell scans are looking for inflammation as well as infection, whereas technetium ciprofloxacin, which is what infecton is a little bit more specific for bacterial infection. So that was the rationale at that time, where we were working on a very specific agent for infection. So anybody who works on radiolabeled antibiotics and so on will be quoting this sort of pivotal work from now on. So thanks to Keith Britton, I became part of that legacy forever. As you mentioned, over time, I've evolved myself because my medical director at the time told me, you have to do things beyond your own department. Otherwise, you'll get bored, you'll get burnt out. So he actually encouraged me to do things beyond the hospital. And I became all these things over time. And just to add some more spice, I dared and volunteered to become member of the fitness to practice panels of the General Medical Council. The so-called the bad people, the people who blame everybody, the people whoever goes to them gets branded, gets stigmatized and so on. So I said, okay, let me be brave enough to see if I can put myself for that. So I did 10 years on that panel. So that exhausted me forever. Nuclear medicine was nothing compared to that, believe me. And then, of course, I've contributed to the Specialty Advisory Committee and so on. So for the students in the audience, this is very important, pivotal test. So when I was a new consultant, we had a new professor of neurosurgery who came in. He's from Germany, and he very much used to PET. And he said, I want PET. And I said, well, I'm sorry, we don't have PET yet. So, but I can give you gamma camera PET. He said, I don't care, give me something. So for people who might not be familiar with this, we actually did gamma camera PET before the PET scanning was available, because our German professor of neurosurgery said he wanted PET. So this is a tumor in the right frontal lobe, and you can see, and I hope people can guess what is the tracer for this one. It's not used now, but it was clinically available in those days. It is thallium-201. So thallium-201 is a very good agent for tumor. So you can see the combination of the FTG and the thallium-201 gave the neurosurgeon an idea to say, okay, what am I going to do? So this is the pre and the post scan. The pre scan is on the top. The post scan is below. And I said to the neurosurgeon in all my excitement, oh, congratulations, you're a neurosurgeon. You've removed it. You've chopped it off. It's all gone. And he said, no. I have not removed it. Because you showed that it is FDG avid, I actually treated the patient with chemotherapy, which was water soluble. So therefore, I could make sure that the tumor could resolve by itself. So very strange for a surgeon not to operate, but very good use of PET scanning even in those days. So I thought I was very impressed with that. And just to show, this is the pre and the post thallium scanning as well. So something to bear in mind, that we did have access to some tracers, but obviously the PET machinery has taken on a completely different role now. And this is the converse. You can see a cold spot. And again, so non-avid on FDG and avid on thallium-201, so a different molecular marker. So even in those days, this is about 20 years ago, we could characterize the brain tumors in this way. So these are some of the papers that we published. This is in 2003. So we can see that, and that's the neurosurgeon at the top, the first author. So it was very, very, very seminal paper, according to me, if people want to know more about functional imaging, much before what's happening today, of course, with all the exotic traces that we see. Over time, I said, okay, we should do something in Liverpool. So I was lucky enough to be the coordinator for the BNMS autumn meeting in 2008. And that coincided with us being the European Capital of Culture in 2008. So the city laid on a lot of extra incentives for people to come. So we got styled as ambassadors for Liverpool because we brought the BNMS to the conference, BNMS conference to the city. So I thought that was very good touch from the city. But I think it was a very good meeting in the end, where we focused on the future of nuclear medicine services. Over time, I found myself supervising students and juniors from a wide range. So some people came with infection as their background, some people came with cancer imaging, some people came with ethics, some people came with radiation risk. So there's a range of applications. And then we go on to theranostics with Y90 dotatate. So this is the next seminal paper that I would like to share that we got involved in, because this is before Lutathera and lutetium came on the scene, where we could access Y90 dotatate and DOTATOC. So this is all before these were licensed agents. This was before Novartis got involved in a big way with Lutathera. And this is one of the papers that we used. And this is something, again, we're quite proud of from Liverpool. So this was published in 2013, you can see. So it's quite an important paper that sets the scene for PRRT in many settings. And again, we were not happy with the conventional way of assessing response to therapy, because, as you know, RECIST criteria is very black and white. So you have to have a shrinkage in the tumor, whereas the neuroendocrine tumor patients are all different. Some people will say, I'm feeling better, but how can I say that I'm feeling better? So this is one of the areas where you can have a clinical improvement, you can have a biochemical improvement, or an imaging improvement, or a combination of all three. So neuroendocrine tumors is one area where your traditional RECIST criteria does not work very well, because it's going to be, you're going to set yourself up for failure if you use just one criteria. Along the journey, the reason I put this up, it will become very obvious very soon. So in around 2016, we talked about thyroid cancer. David and Sabina have talked about having patient contact in clinic. So we are one of the departments where we have thyroid clinics twice a week. We still maintain contact with patients. And the reason I put this paper up specifically is when I visited China, there was one person who was not letting go of my passport. And I thought, this is something dodgy behavior. Why is somebody not letting go of my passport? And then later on, she comes to me and says, do you know your date of birth and my date of birth are exactly the same? And she is a professor of nuclear medicine in Beijing, and I'm in Liverpool, of course. So, can you imagine two people born in two different parts of the world at the same time or on the same day? So we then figured out that she's older than me by 4 hours. So she's now my elder sister, and I'm a younger brother. So she sends me messages every year. So very, very interesting that, you can come across people who have been in different parts of the world, born in different parts of the world on the same day and end up on a similar career path. Very, very strange, very, very unbelievable. I know people might recognize the picture behind. This is Petra in Jordan. So again, we've been on courses where people have interacted from different parts of the world. And you can see this, we've also got journals called the World Journal of Nuclear Medicine, which we also want to promote because that has got a very good global reach. So we should be able to present there as well. So this is one of the reasons I put that up. So one of our trainees who came from Thailand did this work. And so you've got PRRT, you've got gallium-68, DOTA peptide scans, and then you pick up this incidental cardiac uptake. And then you say, well, what does that mean? So does that mean that the patients are going to do better or worse, or we don't know? So we gathered a small series of these patients and have been following them up over time. So hopefully they'll be doing better. We'll have some data to report soon. But something interesting to bear in mind that just because you see hotspots all over and you select for PRRT, that's fine. But then the additional cardiac lesions, nobody's sure of even till this date, but hopefully we'll get some answers soon. So you can see cardiac infiltration. The reason I put the PET panc study here is because we got our PET scanner on the back of a research trial where our pancreatic surgeon said we need to get PET evaluation in PET panc in pancreatic cancer. So people believe that the evidence for pancreatic cancer and PET CT is low, but that's what we wanted to answer, and that's how we got our PET scanner. So my involvement from a clinical NHS point of view was, well, do we pay the VAT so that we can study patients on it or not, or whether we keep it as a simple R&D tool so that it'll never be available for patient use. So luckily we fell down on the side where we did pay the VAT so that we are able to use it for patients as well. So that's one of the important pivotal moments that allowed us to access a PET scanner much before other centers. So this is one of the ethics questions that I raised. Do we need a universal code? This has been taken up in the European Association for further understanding. Communication of radiation risk is something which is very crucial for me because, what do we mean when we say likely to be metastatic or could be metastatic or no definite evidence? All these have got some mathematics associated with it. Curriculum development, again, we have been involved in some work with the IAEA on this matter. And image quality, you can see we've done some JAFROC studies with the Salford group, Salford University. So building on to David Keeling's point about adverse reactions. So one of my legacy roles that came was I get notification every time somebody fills an adverse event on the BNMS adverse event form. So we then use this as publication in 2018. So something to bear in mind that we do look at this every now and then. Quality assurance of reporting much before all of what's happening now. We talked about it in 2010. And role expansion. The radiographers, technologists want to always talk about role expansion. Are they happy? Keep them happy. So we want to keep them happy as well. So just last couple of slides on osteomyelitis. We did not have access to white cell scans because of problems with our radiopharmacy. So we started doing PET CT. And then our orthopedic surgeons got very excited because they say, well, you got some tools which are better than CT or MR in the correct clinical context. So we are using routinely now PET CT for diagnosis and localization, and have almost replaced MR or CT. The negative predictive value is very good. and the positive predictive values, of course, to be very tested soon. I started working with the IAEA on Quantum, which is a quality management of nuclear medicine, and we published on assessing of nuclear medicine practices, which is quality improvement. And again, we've got Jimmy Bomanji and people from the IAEA contributing. Coming to the last set is about interaction with EANM. I know Sabina is quite strong on interaction with EANM. So I was one of the initially the UK delegate to EANM, and now I'm a member of the Ethics Council. I tried to get EANM to host the annual meeting in UK during my presidency, and to see if Liverpool could be encouraged to give them a good deal, but somehow it didn't materialize. I also tried to negotiate a linked membership between BNMS and EANM, much like UK INETs and ENETs, where if you pay to one, you automatically become member of the other. But again, that didn't work out. So tried to do it, but these are the things which didn't work out. So proud moments, comprehensive coverage of nuclear medicine, diagnostics, and therapeutics. We do a bit of everything. We are a big center for brain imaging for dementias, PET-CT for sarcoidosis and orthopedic infections. Personally, I was given one of the Oration Awards from India in 2017 and the President's Medal in 2020, which is quite a proud moment for me because it values my recognition, recognized from both my two constituencies. One is India and the UK. So I'm very proud of that. So this precedes John Buscombe. So he's not there because he was not yet a president. He's not yet a past president. So this is the 50th year photograph that we took. So I had all these illustrious presidents before me. So very proud moment where most of the people behind me with me were my teachers. So very, very proud moment. So in summary, long personal journey in nuclear medicine in the UK. Always believe in onwards and upwards. Thank you very much.
Thank you, Sobhan, for a fabulous journey and for sharing this with us and also highlighting nuclear medicine developments along with your journey. It was really rich and successful, as you said, onwards and upwards. And this is for our 60th anniversary. We are having a new photo. So David, I could see you were there, and John, you will be within past presidents. So yeah, we're looking forward for the next talk. Last but certainly not least, it's my pleasure to introduce our final past president this evening, Professor John Buscombe, who served as the BNMS president from 2018 to 2021. You may notice that his term was longer than that of any other president, and John will remind us why. So I just also want to tell you that we can actually go past 7 o'clock. So to ensure to hear fully from the past presidents, so please bear with us if you have questions after John's talk. So we'll discuss further. So, and we have extension for this webinar for the next 15 minutes. So basically we can stay until 7:15 or even longer. So John is a leading figure in the field of nuclear medicine in the world and in the UK, of course, and a long-standing contributor to development of nuclear medicine. He retired at the end of 2025, sadly for us, but I think he will probably continue in some way, although he says he wouldn't, but I know him, he will. He will continue with his teaching. If anything, his YouTube videos are most popular videos on our channels. And I think when John gives a lecture, that's always well received. So John was a professor at the University of Pretoria, and he retired from his consultant job in nuclear medicine, Cambridge University Hospitals, and later on he works at Barts as well. He trained in medicine in London Hospital Medical College, graduating in 1984. Completed his training in general internal medicine first before specializing in nuclear medicine at Middlesex. And also he was appointed as a consultant in nuclear medicine at Royal Free Hospital when David was a president in 1994. And during this time, he pioneered the concept of dedicated diagnostic services and helped build one of the UK's leading radionuclide therapy centres with a particular focus on the imaging and treatment of neuroendocrine tumours. John has extensive experience in both diagnostic and therapeutic nuclear medicine. Most recently, he was one of the founding contributors to the MSc PGD PG Cert postgraduate program in nuclear medicine at the Brighton Sussex Medical School, where he masterminded the molecular radiotherapy and diagnostics module. He has published more than 250 peer-reviewed papers, authored 8 textbooks, and has been instrumental in shaping education in our field. This evening, John will present his talk on molecular radiotherapy to infinity and beyond. Before the talk, I want to ask John similar questions about what challenges and how he can explain his 1 year longer term or even more too. John? 1, okay. What was the reason? What was your main key challenge during your presidency?
Well, to be honest, it was meant to follow on from Sobhan's work on Brexit. So I sort of rearranged my contract at Cambridge to give me free time to do all the work that would be required for Brexit in preparation, particularly for the delivery of radiopharmaceuticals, which involved multiple meetings at the Department of Health, etc., going to House of Commons, House of Lords committees, trying to sort of convince people that need to take it seriously, following on for the excellent work that was started by Sobhan. That all sort of seemed to be going along very nicely, and when COVID struck, and to be honest, it was a bit of a challenge to us as a society. We had tried to book ahead 2 conference centres, and we didn't have much money in the bank, and we suddenly realised there would be no conferences. At that time there was no. We weren't going to be told we're going to get any compensation. Nothing was going to happen, and we really needed to do a lot of work to just try and get the society to survive. And Charlotte and Caroline in the office and Gilly as treasurer basically did most of the hard work on that. So we should all be grateful to them for really changing the society to one that would actually survive all that happened with COVID. And as a consequence, because my successor was trying to deal with COVID in his own hospital, we had to have an extraordinary annual general meeting so that my term could be extended a year until such time as the sort of COVID became more manageable. So yes, interesting times.
It was very difficult, was it? And then thank you all for working so hard to, taking us through this most difficult period. What actually inspired you to take on this role as a president?
Well, I was a bit like David. I was a bit pushed by various people who said, you're basically getting on a bit. You could do something useful for other people now. So, and I think that there was some concern that the Brexit situation actually proved not to be as bad as we thought it would be, but it could be quite severe. So, as I said, I was in the position where I could sort of basically do a retire and return from where I was working, so that I could then have much more flexible working time if I needed to take a day off to be at a meeting or a conference or go to Brussels, so it enabled me to do all those things without, which I think would have been much more difficult for somebody who is still stuck trying to fulfill a complete job plan.
Yeah, absolutely. I think you were a definite figurehead of BNMS and led us through this difficult period. And what was your most rewarding, memorable moment of your term then?
I think really how everybody pulled together, particularly over COVID. And I mean, Sobhan was brilliant. You were brilliant. Lots of other people probably listening were also brilliant, either locally, and they gave me lots of stories of how they managed to cope locally, finding local solutions to make sure that they could get a service up and running again, and they could do that safely, and that patients would have confidence in it. So I think my proudest moment really is that the BNMS, at the time when it was at its most stressed and faced the most challenges, rose to those challenges, and people who worked in nuclear medicine really showed their professionalism and their dedication to both their profession and also to their patients.
Fantastic, John. And any development that you could recall that you did, you really tried to enable access to patients for nuclear medicine, but for theranostics in particular.
Yes, I mean, towards the end of my presidency, we were able to set up what was meant to be a small committee, but ended up having to work mainly online, which actually wasn't a bad way of working, actually. So we, jointly with the Royal College of Physicians, the Royal College of Radiologists, Radiotherapy Section, and IPEM, we put together a sort of report of the status of nuclear medicine across the country. And I got a couple of slides of that in my presentation, and put together a report of suggestions of how we could move forward. And that's sort of evolved into the Molecular Radiotherapy Consortium, which is still working. And I think the most important thing is to work together. So we work very closely with our clinical oncology colleagues and with our medical physics colleagues, as well as those interested parties, to try and ensure that people could access nuclear medicine therapies actually where they were, instead of the thing that happened when I was younger, where patients were traveling 6, 7, 8 hours to get treatment.
Yeah, absolutely fantastic work, John, and thank you so much for enabling patients to access this targeted treatment, which is truly advanced and personalized. And as you know, this document is still the only published document, and clearly now we really need to build up again on your work, and that was a definite legacy that you left for us to do for the future as well. So I think we'll move to your talk now, please, Molecular Radiotherapy to Infinity and Beyond, and we'll come back to questions from audience. Thank you, John. And I just want to say, before you go with the talk, I really congratulate you on your retirement, and I look forward to seeing what you're doing next.
Thank you very much. I think Tom is going to very kindly put my slides up. So I'm just going to say next slide. So here we're just going to have a little review of molecular radiotherapy, of its evolution and where we're moving forwards to. So next slide, please. So I don't have any disclosures. Nobody's interested in anything I say from a commercial point of view, which is great, really. I'm going to talk a little bit about our origins, the rise of theragnostics, the situation it was when we did the review in 2020 and 2021, then to infinity and beyond. Next slide. So our story is 89 years old. BNMS is 60 years old, but 29 years before that, Saul Hertz, an endocrinologist in Boston, who had a really good understanding of thyroid physiology and pathology. It's very interesting that David also came from a background of pathology. And I think that really ties in. If you understand what goes wrong and how it goes wrong, I think it's a really good clue of how you can then image it and treat it. So in 1936, he had the idea of using radioactive iodine, started off in animals, and he talked to Carl Compton. We know Compton age fame, and he produced a small amount of iodine-128 for work in rabbits. Next slide. So the breakthrough came, he got some funding from something called the Markov Foundation in New York. He built a cyclotron with his funding. It's a bit like the story from the Hammersmith, just get on with stuff. And that was able to make a mixture of iodine-130 and iodine-131. And in 1941, the first patient was treated, and patients were treated with both overactive thyroid and with thyroid cancer. And to work out how much to give, he would use a smaller activity, measure that in the measuring system, and then use that diagnostics to produce a therapy. And that really is the basis of theragnostics. It's the knowledge that we get from the diagnostic, which then helps us with the therapy. Next slide. So here you have a picture of Saul Hertz with his measuring machine. There were no gamma cameras. There was nothing like that. What he could do, though, is he could measure the amount of uptake of the radioiodine. And here's a picture with the measuring system. Now, the person sitting in the chair is actually his personal assistant. Now, those of you who are sharp eye will notice that she's actually facing the wrong way. This is because evidently she had her hair done, and she wasn't going to have people taking the picture of the back of her head when she'd taken so much effort to get her hair looking nice. So she sat in the chair the wrong way round, but she should be sitting the other way. And here you see Saul Hertz with his machine. Now, why do I say theragnostics? As you know, it's a bit of a bugbear of mine, because In Greek, theros is to treat, and gnostics with a g is to know. So you put the two together, you know how to treat therognostics. So it's just a little thing I have. Next slide. We move on to another endocrinologist, Eric Krenin, and his team over in Rotterdam, working with a series of chemists in Basel in Switzerland, and also with a group of clinicians, which actually included Keith Britton and Jimmy Bamanji, developing a method of imaging somatostatin receptors. Now, these were important, or been found to be important, in neuroendocrine tumors, an endocrine tumor that was rarer than thyroid cancer, but patients had a pretty miserable life because they didn't have any effective treatment. Chemotherapy didn't work. And therefore, Eric Krenin had the idea that we could image these tumors by using the somatostatin receptors, particularly subtype 2, and therefore developed indium octreotide. Now, they tried it with iodine first. It didn't work very well, but the indium with the DTPA linker worked perfectly. His first case he published was in 1990, and by 1993, he was able to publish over 1,000 cases. Now, that is a lot of cases. And he could find 90% of these neuroendocrine tumors, even some of the more malignant ones he could find as well. The only ones he wasn't so good at finding was insulinomas. We know that as a mixture of the fact they don't have the right receptors, and also they're quite small, and we needed to move on to PET to find those. Since around the 2000s, we've used SPECT-CT for that. So I think here we have a SPECT study which shows two blobs and what looks like a colon. But I think we do the next slide. You can see that when you do a SPECT-CT, they're not a colon. It is in flat peritoneal metastasis. And I think that Sobhan clearly showed that the quality of the imaging helps us to get the gnosis to do the therapy. Next slide. Now, this moved on to develop treatment. We started, first of all, with just high activity indium. Then we moved to yttrium, and then to lutetium. And here we have one of the first reports of the lutetium dotatate. And you can see here on the graph that as long as the tumor didn't progress, which is the yellow curve, it didn't really matter if it got smaller or stayed the same size. Patients' progression-free survival was the same. And again, we moved forwards on our imaging so that we were imaging patients with both SPECT-CT, and using gallium dotatate or DOTATOC, we could then use PET-CT as well. Next slide. This eventually became the NETA-1 trial, and this was developed with Novartis, working with centres across the United Kingdom, across Europe, North America, and Australia. And this provided a proper phase 3 randomized controlled trial, clearly demonstrating that treatment with lutetium dotatate improved your progression-free survival as against just having high-dose, unlabeled octreotide. Now, this is one of the new ways that we have to do things in nuclear medicine, and particularly in therapy. We now have to do phase 3 trials to prove it works compared to conventional treatments. And this shows very clearly the advantage of giving these patients the dotatate. Next slide. So the next target, as we all know, is the wonderfully named glutamate carboxypeptidase 2, which is a transmembrane glycoprotein, which is involved in bringing folate into the cancer cell. But we all know it as prostate-specific membrane antigen, or PSMA. And this is a little cartoon of the molecule which goes across the membrane. And there are multiple targets on here, but most of the targets that we use using the ligands that are available are in the external part of this molecule. And here we have a patient from St. Bartholomew's, and here you have a gallium-68 PSMA scan, and you can see the classical normal distribution. And in addition, if you're very get quite close to the screen, you can see that there are two little blobs at the back of the prostate, and one extends right into one of the seminal vesicles. So you can stage these patients very, very accurately with this imaging technique. Next slide. But obviously, we're interested in theragnostics and treatment. So this moved very rapidly on to a phase 3 trial. So whilst it took 20 years from Eric Cranion's idea to get to a phase 3 trial, it was less than about 5 years to go from PSMA PET imaging to a trial starting and it reported 2 or 3 years later. So we've taken about a decade off that process. So where people can start to see an advantage, then things start to move forwards. Now, this is the results of what's called the VISION trial. And this compared best supportive care versus lutetium PSMA in patients with a castrate-resistant prostate cancer that's metastatic. And you can see here an overall survival. There's an increase of about 11 months in overall survival in those patients who received lutetium PSMA versus conventional treatments. Next slide. So we knew that this was a common cancer, and whilst neuroendocrine tumors, patients could go to a few centers of excellence, this wouldn't work for PSMA theragnostics if it was actually going to occur. So this is why we've developed the idea of having this review of molecular radiotherapy across the United Kingdom. And this was this joint program that we did with the RCP, RCR, and IPEM. And we looked at the provision of presently funded MRT, so that we picked 3 things that was funded by NICE, which was the lutetium dotatate, radium-223, and SERT. And this exposed very, very deep regional differences. So this is a map of the United Kingdom. So those where within 50% of the average was being delivered per million population, they're green. If it's between a quarter and half, it's orange. If it's less than a quarter, it's red. And if we didn't get any information, it's Northern Ireland. Now, Northern Ireland, we didn't know what was happening. This is actually with radium. What we didn't know was, in fact, they were doing very well in Northern Ireland. They just didn't like answering questions and filling in questionnaires, but they were doing fine. But what you can see here is if you were unfortunate enough to live in Yorkshire or Humber, your access to radium-223, which is a really easy treatment to give, our nurses give it. Okay, and it's really easy to do. It doesn't require any expensive equipment. It just requires some training and the will to do it. Your chance of getting it was nearly 1/10 of what it would be if you lived in the southwest of England, which had the highest delivery. So well done, David. Your successors were delivering the highest number of radium-223s across the whole of the UK. So this exposed deep divisions, and we found that this was unfair, that whether or not you got molecular radiotherapy really depended where you lived. And so we produced a report, which we sent to all the national governments to say, look, guys, you've got to do something about this. So next slide. So we sent this off, and it was all beginning to look as though they were going to take it seriously. England obviously took slower. Scotland was very good. Wales were pretty good. Northern Ireland didn't answer, but then they finally got a government and started to sort things out. But then we got NICE the bogeyman. So it was all going forwards. We'd done the trial. We had patients who were being treated, partly funded by the NHS, partly funded by Novartis. Then the VISION trial went to NICE, and NICE just said it wasn't value for money and they weren't going to fund it. So there is an impasse at the moment, as we all know, for those of us who are very frustrated that we can't treat patients we know would benefit unless they're on a clinical trial. But we know that this won't last forever. And the reason is there will be other PSMAs that come available. They may, as they compete, the prices will drop. They will get below the barrier that NICE will accept. And I'm sure they will fund at least 1 or 2 of these agents. So it's still coming. It's only a temporary halt. So don't think I'm not going to be doing this in the future, because you will. Okay, next slide. But life moves on. Now, despite the problems with funding, molecular radiotherapy research continues apace. Okay, we cannot stop this. You may want it to stop, but it can't be stopped. When we did the review, we found 16 molecular radiotherapy phase 2 and 3. We ignored phase 1, phase 2 and 3 registered trials on all the clinical trials networks. When I looked at the end of last year, there's over 400. Okay, all in different agents, all with different cancers. Last year, $6 billion was invested. Last 3 years, $6 billion invested in molecular radiotherapy. Now, these companies aren't charities. They will not be investing money unless they think that this is actually going to be the future. Now, if only 10% of those 400 agents are successful, and that's about the average, that means that in the 10 years' time, nuclear medicine will be working with 40 new agents, possibly in 40 different cancers than they're doing at the moment. So there is infinity and beyond, and we just have to be ready for it. Next slide. I think. So new ideas, well, we can mine radioisotopes. We can get lead-212, which is an excellent isotope. And you can see below where 212 has been used in treating patients who have prostate cancer metastases, and you get very good clearance. Terbium-166 with Auger electrons can find micrometastases, so we can treat micrometastasis with that, so we can use it as an adjuvant therapy in patients who don't have a positive PET scan, but we know may have small amounts of tumor. And also we can direct radiopharmaceuticals to particular sites. So the bottom is a patient that was treated in Addenbrooke's in Cambridge, and you can see here there's a patient with an surgically inoperable pancreatic cancer where a phosphorus-32 gel has been injected by EUS. Notice all this means we work with other people. And you can see there's a significant reduction in the FDG-avid tumor. Just the start, but you can see that different approaches enable us to treat different cancers. Next slide. So these are some of the heroes of diagnostics. Some of these people you may recognize. Some of them whose papers you may have read, and in the middle of that is our founder, Saul Hertz, who really gave us the idea. And some of these are chemists, some are medics, some are patients, but they've all driven the theragnostic vision, and that vision is growing year by year. Next slide. So, in summary, the next 10 years may be the most exciting in nuclear medicine. It's been pretty exciting so far, but it may get even more exciting. And for those of us who were like Sobhan and myself, who started off with theragnostics and nobody really wanted to help us, everybody's going to be doing theragnostics. Even if you don't want to be doing it, you're going to be doing it. And there is an infinite combination of radionuclides and targeting molecules. Many of these won't work, but a lot of them will work. So there's big changes in the next decade. And beyond that, who knows where we'll be? That's up to you guys, not to me because I've retired. Thank you. You can finish sharing the slides.
Thank you very much. That was fabulous talk, John. And as you said, diagnostics is non-stoppable. The same happened with the PET 2 decades ago. It's something that, it's a major breakthrough for cancer treatment together with the potentially vaccines and immunotherapy. So you answered my question about what is your vision for the future of nuclear medicine over the next decade. And I think we go now to questions from the audience. So I think going back to questions for the presidents, it's Alan Perkins is saying, thank you all. It's fascinating hearing the histories of our past presidents in the development of our specialty. Looking to the future, how should the BNMS ensure that it remains significant in promoting the specialty? So who is going to take this question? John?
Okay, I'll say that. I mean, I think it's an evolution, not a revolution, but I think that multiple presidents have tried to work with other societies. We often have joint meetings. The ENM has been a big leader on this. So, for example, in the ENM, almost every single set of guidelines now works with a clinical society. And I think that the two things I think is important is to work with relevant clinical societies and specialists, and also with patient advocacy groups, because they actually really help with funding. So it's a patient advocacy group that got CERT funded for HCC, not medics, patients. Yeah. So that's what we need to do. Continue to cooperate and work with other people.
Excellent. And I think I already proposed that like we add as advocacy as a aim of the BNMS that's going to go through the next council meeting. I mean, we're already doing it, promoting and actually working more with the patients as well. So I think that's something that we are going to build on and build up. So I think there was a question for David, because that was written at the time that David finished, although he did, I think, probably answered that question already, but I will repeat. Maybe he might have some other ideas to talk to us about. What was the biggest step change in nuclear medicine, technological or clinical, during your career?
The advance in radiopharmaceuticals. Our machinery has advanced, yes, but the big advances come from new radiopharmaceuticals, new approaches. That's why I've been interested to hear about this evening. My first tomo scanner was a rectilinear tomo scanner. I mean, this is archaic today. But we could at least go round ahead, and Andrew Todd Pokrapek, who you ought to know, he would work away on the computer, having taken it down on fast punch tape. This sounds like sort of 18th century work, but it isn't. It's in my lifetime.
Oh, it's like not long ago when you consider what actual development really happened during this period. As you said, in your period, probably radiopharmaceuticals, but then we have a huge technology developments. Now, again, we have again radiopharmaceuticals developments. So I think, yeah, I definitely know Andrew Ted Pokrapek because he was one of my examiners for MSc. So like I know like that area of nuclear medicine and physics involvement was also very, very important, is it, at that time? So anything else that you want to tell us, David? Could you give advice? What would you offer to next generation of professionals entering the field today?
I think you've got to be a slightly different doctor from the, if you like, the GP material. I started off wanting to do something on the scientific side, which I thought in 1959, when I qualified, meant becoming a pathologist and researching there. But in those days, the pathologists, the only time we saw patients was running the anticoagulant clinic. Getting blood out of Churchill was an absolute bloody nightmare. But that was the sort of job we were doing. And it was the move to nuclear medicine where we started seeing new things and handling patients, looking after them. That's what appealed to me. And I grant you, I'm not your average GP material doctor, but those of us in nuclear medicine, I think we find ourselves drifting into it, whether we like it or not, almost.
Absolutely, I agree with you. It's like even if you have the same birthday at a different time, part of the world, we find each other somehow.
Yes.
We do have a similar interest in a science and medicine and the outcomes of patients. So thank you, David. And Glenn Flux, comment and question. Thanks for the brilliant review and all of your work to all hosts and panelists. The wave of radiotherapeutics will cost a fortune at commercial prices. Do you think this block can be mitigated by the nuclear medicine imaging, dosimetry, early administration in the pathways? Sobhan?
Yeah. I think what following on from what John said, there are going to be big players and numerous number of players coming in. So that's going to automatically introduce a bit of competition in the system, whereby people will have to look at their prices. Companies will want to get their investment back. And let's face it, prostate and breast cancer are the two biggest bills for any company that wants to develop cancer work. So the company that invests more in anything that is related to prostate and breast will get their money back in a bigger way in these areas. In the rarer areas, rarer tumors, neuroendocrine and all, they are just a way to get into the bigger market. So neuroendocrine tumors, I mean, I'm actually pleased that Novartis has invested heavily in this, because, if we didn't have the commercial backing, we would still be struggling with an unlicensed product. So, but Novartis saw that as a way, saw Lutathera as a way in to get into this theranostics concepts. And I think the big companies will aim for big bill clinical areas as well. I think with regards to dosimetry and earlier administration, obviously we should be incorporating all good practices into our systems, and the cost of that is not going to be that much in the grand scheme of things if we get it right. But we need to get the commercial partners back on side. We need to get patient advocacy groups, because as John mentioned, patients are the biggest group that will back us. And I think it is possible, but the biggest motivation, I think, is going to be sheer money. People want to make money, but there are going to be many players coming in, not just one big player, and that's going to drive down the cost.
Yeah, I think competition will drive costs down, but earlier treatment is certainly something that we need to aim on.
Sure.
Treatment at early stage in the patient's pathways, and fortunately we have now trials when we are putting PSMA at like hormone sensitive, prostate cancer patients, and I think that will be definite way how to get approval through NICE as well. So should I just say that final remarks, like as we celebrate 60 years of the BNMS, any final reflection or messages you'd like to share with the BNMS community as we mark this milestone? John?
Well, I think that those of you who are working in nuclear medicine now are going to see that the next 60 years are going to be a lot more exciting than the last 60 years, and they were pretty exciting.
Absolutely. Never better time for nuclear medicine. Sobhan?
Yeah, I mean, I would say believe in yourself, have faith in your beliefs, trust your instinct. If you're coming into it as a medic, as a physicist, trust your core instinct. Support BNMS, because BNMS is your champion. Join BNMS Council, take up one of the officer posts. Don't just stand outside and say, yeah, they do it, don't they? Become part of the change. And, I think it's a lot more welcoming than people realize.
Get involved. Thank you. Thanks, Sobhan. And David, anything that you could wish us for our 60th anniversary, and as we celebrate this mark this milestone, any reflection or message?
I'm very happily proud that people like Keith Britton and Peter L. started the nuclear medicine in my department. And I seem to have set them off well.
Oh, lovely.
They're doing very well, thank you.
Thank you so much. We did invite them as well to contribute and to come for the award ceremony, so they will respond in their own ways, and I think Keith Britton will certainly share his message online. And it was a real pleasure to have you all three today, and I think, David, it's a remarkable journey. We just all really hope that,
still alive.
It's amazing, you're just fantastic. And John, happy retirement, and we'll, and Sobhan, we'll work together, build up for the future. And thank you all. And please join our next webinar, which is with the female presidents next time in February. And I think we'll have a really successful discussion next time with the female presidents too, as we had with our first webinar today. Thank you all, and have a lovely evening. Thank you.
Thank you.
Bye.
Thank you.
Bye-bye.
Bye.